What if I told you that the instruction manual for building you, every cell, every thought, every beat of your heart, was almost completely unreadable? That's the bizarre puzzle that has haunted scientists for decades. When the Human Genome Project finished its monumental task in the early 2000s, the world waited for the book of life to be opened. We expected tens of thousands, maybe hundreds of thousands of genes. What we found was a shock. The genes, the parts of our DNA that code for the proteins that build and run our bodies, made up a tiny, almost laughable 2% of the entire sequence. The other 98%, it appeared to be nothing. A vast, dark, repetitive desert of code. We gave it a dismissive and frankly arrogant name, junk DNA. We thought we were carrying around billions of years of useless evolutionary baggage, like a hard drive cluttered with deleted files. But we were wrong, so incredibly wrong. The story of what's really hiding in that junk is far more strange and beautiful than we ever imagined. It's the story of how science learned to see the wilderness for the trees. The first clue that we were missing something big came when we tried to understand life's greatest magic trick, how a single fertilized egg can build an entire human. All our cells, skin, heart, brain, have the exact same genes, the same DNA blueprint. So how does a brain cell know to be a brain cell and not a skin cell? Why doesn't your eyeball start trying to pump blood? The answer wasn't in the genes themselves, but in the junk surrounding them. Imagine your genes are the musicians in a massive orchestra, each one a violin, a trumpet, a drum, ready to play its note. The junk, DNA, it's the conductor. This conductor doesn't play an instrument, but it decides who plays, when they play, and how loudly. These regulatory sequences with names like promoters, enhancers, and silencers are the grand composers of life. An enhancer region can be thousands of letters away from a gene, but it acts like a dramatic wave from the conductor, telling a section to play with passion, boosting its activity tenfold. A silencer does the opposite, hushing a gene that's not needed. This is what allows for the stunning diversity of life from the same basic set of genes. The incredible variety we see in human faces, for instance, is largely due to tiny, subtle changes in this regulatory DNA, not the protein-coding genes themselves. It's the conductor changing the performance, not the musicians changing their instruments. But the conductor wasn't the only secret hiding in the dark. For decades, we believed in a simple process. DNA makes RNA, and RNA makes protein. The RNA was just the messenger, a dutiful courier carrying the recipe from the DNA blueprint to the cell's protein factory. Then we found a whole other class of RNA that never becomes a protein. These molecules are transcribed from the junk regions, and then they go to work. They are the hidden workforce. Think of them as the stage crew, the security team, and the quality control managers of the cell. One type, called a microRNA, is like a tiny, programmable assassin. It's created from a snippet of junk DNA, and its sole purpose is to hunt down a specific messenger, RNA, the courier, and shred it before it can deliver its message. It's a stunningly precise way to control exactly how much of a protein gets made. This isn't junk. This is a microscopic system of checks and balances. And then there's the wildest part. Hiding in our DNA are ancient renegades, pieces of code that are, in a way, still alive. They make up nearly half of our entire genome. We call them jumping genes, or transposons. For decades, the leading theory was that they were ancient viral infections, selfish DNA that only cared about making more of itself, a squatter living in our genome rent-free for millions of years. And sometimes they do cause chaos, but they are also a source of incredible creativity. Every time one of these genes jumps, it's like a genetic lottery ticket. It might land harmlessly in a vast stretch of nothing. It might land in the middle of a critical gene, breaking it and causing disease. Or just maybe, it might land next to a silent gene and accidentally turn it on. It might create a new instruction for our orchestra conductor. Over millions of years, these happy accidents are a major engine of evolution, creating new traits out of thin air. Finally, some of the junk really is a graveyard. It's a ghostly fossil record of our deep past, littered with the broken remains of genes that worked perfectly in our distant ancestors but are now silent. These are called pseudogenes. Inside every one of us is the ghost of a gene. It's the final piece of machinery needed to make vitamin C from scratch. It works perfectly in most mammals, from dogs to cows. But somewhere about 60 million years ago, in a primate ancestor of ours, a mutation broke it. The gene died, but its corpse is still here. copied and passed down through millions of generations, a permanent reminder in our code that we are forever tied to a world where we must find our vitamin C in fruit. So, this junk DNA, it's not junk at all. It's the conductor of our genetic symphony, the stage crew that keeps the show running, the chaotic artist that drives innovation, and the dusty, fossil-filled library of our entire evolutionary history. The 2% of our DNA that makes proteins are the storybooks. The other 98% is the library itself, the architecture, the card catalog, the librarians, and all the forgotten volumes in the basement. The biggest breakthroughs in medicine in the next century might not come from the 2% we thought we understood, but from finally learning the language of the 98% we once so arrogantly dismissed.